recap. We want to predict in which subcellular localization a protein is. Actually, why would we want to predict that? Proteins move around in the cell. Why do I want to predict one subcellular localization? Don't know.
Proteins have one specific function and one compartment.
Yes, of course you're a little bit biased on this one, but you're totally right. True. So, in fact, it turns out that you may call that surprising, or you may have any sort of word to describe it, but the vast majority, no, not the vast majority, but most proteins clearly, in fact, are native to one particular compartment. Native means that the function that we typically associate with those proteins happens by them being in one particular compartment. And that particular compartment is sort of ingrained somewhere on the sequence or somewhere, at least on its sort of historical path. Now, ingrained, it can be on the sequence. It could be a motif, it could be something that is sort of average amino acid features, something that makes it fit into a particular environment. What else could be the reason for ending up in one particular compartment for a protein? So there's nothing ingrained in that sequence. How could it still end up in the right compartment? Any idea? Nobody? Yeah. Nobody? Nobody other than Natalie? And I'm understanding that Natalie currently also is not saying anything, but you said something already. No? How do you believe could a protein get to a particular place if it's not sort of directly written on its sequence where to go to? Yeah? Vanessa?
Vesicles?
Yes. Now that sort of is so you're right. So you could have, in fact, I believe your solution is in the right direction. Now, vesicles, typically when we say the word vesicles, we assume or we imagine little compartments that are membrane bound. So we are thinking about sort of subcellular compartments in some sense, right? And then the question would be, how do I get into that subcellular compartment in some sense? There is a vesicle targeting site, again on the sequence, but my postulate here was something that does not have any of these targeting sites. But essentially with the vesicle idea, you're on the right track. It's not quite a vesicle, but you're on the right track. Yeah?
Maybe also the size of the protein.
Oh, so the sorting mechanism by which the big ones go.
For
Okay.
example, the nucleus, there are some pores, and not every protein can go through.
It is so. Now you say something. So in fact, it's gated, and these gates are not. So titin doesn't get into the nucleus. Okay, because titin doesn't get in, you can postulate that very big proteins don't get in because the biggest one doesn't get in. So I proved my example already for one case, right? And since the gates are of limited size, it could well be that the transport is regulated in that sense. It is, in fact. So, Kyla, I have to admit, I cannot remember the distribution of length of proteins between different compartments. What I do remember is that there is a tiny difference. There is a difference between different compartments, the length in different compartments. But I cannot remember how so there are two things I cannot remember. First, how much would I believe it works better than random, but how much better than random, I don't know. So you're just using the length as a predictor. And I also cannot remember if nucleus is the smallest one or not. So absolutely don't remember that. But it's also true, so back to what Vanessa said, so some of these tiny vesicles also have sort of difficult to get into. Mitochondria have two membranes. It's also not that easy to get into. So you could somehow make that point for different things. No, it's not what I meant. So, but it's an interesting idea. So the way I believe I see your answer now is that you, at least Kyla, or maybe others in the group, went off into answering the question by thinking what other feature in the sequence could be relevant for me to be sorted into different compartments. This is not what I meant. What I meant is, in fact, imagine there is no feature that you can actually find. So whatever you do, you look at the sequence, you cannot pick it up. Why could the protein still end up in the right place?
Maybe there is a specific shape of the structure where a ligand can bind.
That's a cool idea. And now we get, yeah,
and the ligand could be some, could cause some sort of transporter to get to the.
That's what I meant. When you started with the 3D structure, I thought you had a different idea. So it could be that a shape is relevant, and that you pick up something on the shape. But Daniel's end of the story is really the one. So you essentially have another protein that binds, right? So you have this green protein that has no imprint for being brought anywhere. But the green protein, in fact, binds to the black protein. And the black protein has a zip code. So the black protein has a nuclear localization signal, for instance. Or the black protein has a signal peptide. So the black protein is brought through the endoplasmic reticulum here, through the endoplasmic reticulum into the outside. Okay? And since it's bound to the green one, it sort of pulls the green one with it. And the green one still, by definition, has no sequence reducible, not size, not hydrophobicity, not nothing. The only way why it's actually secreted is because it's piggybacking. Okay? And we assume that some proteins are, in fact, brought to the right place through this process. What we don't know is what the fraction of these is. Now, in a more generic story, there are very, very different types of protein functions related. Why am I going? I'm going too far back here. What we talked about is that we can predict subcellular localization through homology-based inference. We talked about text similarity, and we talked about motifs, right? So text analysis, and that is again work from Rajesh Nair. The idea is very simply the following. If you removed the word nuclear here, in SWISS-PROT you could no longer identify that this protein is nuclear. Well, actually you can. How?
It is involved in cell cycle regulation and DNA replication.
Exactly. So we have actually keywords in here that immediately give away that this is a nuclear protein, right? Even if you remove this column, you still have. Now, you do sort of need to have some background knowledge. You do need to somehow know that DNA binding is nuclear, that chromatin regulator is nuclear, that something like such as protein and coagulation would be extracellular. But ultimately the idea is, could we not from these keywords sort of grind up these keywords and get a prediction? And in this prediction we want to do two things. On the one hand, we want to essentially find what is trivial for an expert, DNA binding, chromatin regulator, transcription factor. It's trivial if you have some background information. For machine learning, that's not trivial. For a machine, that's not trivial. So you want to sort of have a method that discovers that. At the same time, you want to see whether there are some keywords in there that are informative, but not alone. It is such that the keyword itself, DNA binding, is clear where it falls. But maybe there are some sort of most subtle correlations that together they could help you to, in fact, predict subcellular localization from those keywords. What would you need? Well, you would have to begin with a bunch of keywords from SWISS-PROT and proteins for subcellular localizations. So in this particular case here, I show 5 different classes. And we started initially from SWISS-PROT with 15,000 proteins, for which the subcellular localization was annotated so that we could sort of set up the system. Right? Julian. And for these, we could essentially extract all these keywords. How would you do that? So mind you, this is not free learning. This is not free text. So the way SWISS-PROT is annotated is that the annotators sort of use an alphabet or a dictionary of words. They try, when they have a new publication that presents something new, they will use new words, but they will try to use as much of this controlled vocabulary as possible. So it's not really on the level of free text, it's more on the level of real keywords that describe something. Okay? Anyway, how would you extract them? Well, I mean, you take them and you sort of take them apart and you have a list of keywords. But are you going to use all of them? What are you not going to use? It's really simple. Exactly. How are, as in the, how would you find them? Well, I mean, you can go through the text and just look up 15,000. You're not going to do that. You're not going to go through 15,000 of these records and find all the words that you want to exclude. So how would you find those? Yeah?
Just count all the words in 50,000 abstracts or entries and say if it is in over 10% of the entries, you don't use it.
This is an interesting idea. The problem, I cannot quite remember, but it could be that DNA binding is, I'm just constructing the example because you say 10%. DNA binding could be close to 10%. So it could be that some of the ones, so I believe what I'm making up the point here, then you will say, okay, I don't use 10, I use 5. And that is in the hack, you're right.
But if DNA binding is 10%, then maybe you don't want to use DNA binding.
Then maybe you do. Maybe you do. Let's go back to the point that I want to make. You do want to find correlations that are trivial for the expert, but not trivial for the machine. Again, maybe 10%. Let's not focus on the 10%. I believe this is, Julian, you got a good idea, and I believe this is one way of starting. I'm just saying be wary about the threshold of that. But there's something where you don't have to be wary about this. That could be what? Something more extreme than 10%. Much more extreme than 10%. The first way in which you exclude a bunch of keywords is, yeah?
You just could use nouns, or nothing else.
Yeah. You could just use nouns, like region.
Some are useless, but
Yeah, but how do you find the ones that are useless? What's the definition for absolutely useless? A very simple definition of absolutely useless.
It's in every entry.
Exactly. So there you can completely, no, every entry is one way of saying it, right? But less sort of demanding than every entry. All you need is that you have a word that is in all sub-cellular localizations. So you have that word in all classes. And you don't need that word, right? You don't need it in all proteins. You don't need it in 10% of the proteins. All you need is that the same thing appears in all the different. I showed you 5 different types of sub-cellular localization that you want to distinguish. If it matches to all 5, it's useless, right? And you don't need a threshold. It's in every class out. And typically the simple ones in the and you will actually find in all of them if you have it in 15,000 records. And then we get, I believe what Julian said, I would call rather refinement on that. But the first step really is very simple. It matches everywhere out. Then you should actually, and that may in this example of DNA that I sort of pushed you with, Julian, in the DNA binding, then you may be interested in the ones that are the other way around. It's a single keyword that always maps to one particular class. those in fact would be the sort of trivial correlations that experts such as Felix, or that you guys have learned enough biology, you guys know chromatin regulation, you know DNA binding in terms of which subcellular localization is that mapped to, right? So those simple ones you also would find on the other extreme by just mapping only one class, blood correlation. The way you then extract these keywords is you build a data set where you have the keywords here of the final list of keywords that are non-trivial, so that map in more than one, possibly this is one way of doing it, that map in more than one subcellular localization and in fewer than all, or in fewer than most of them, or whatever you put. And then you essentially have a 0 or 1 depending on whether or not you find that keyword. Okay, now you have the issue. You build this experimental vector database. In this experimental vector database, where you know for all the proteins that you have labeled what exactly are the keywords that come, and you know them for the different types of subcellular localizations. Now you come in with a new protein. How are you going to find which one that correlates to? How are you going to sort it? So my new protein, I have an annotation of keywords, that's the idea here. I do not know the subcellular localization, but I in fact have this vector. Not this one, because that's my database. So that's my experimental database, that's what I want to sort of retrieve. So how am I going to find for a new vector that is not identical to any of those? How am I going to find which subcellular localization that is? Again, we have thrown away the simple ones, meaning DNA binding, blood coagulation in this example, chromatin modulation. So we have thrown the ones away where it's very clear where they fall. And we only have now left keywords that, in fact, none of them is enough, but somehow together they may stand for a particular class. And I come in with a new vector. What do I do? Is this difficult or is it easy?
There's like a difference between those vectors and my new vector.
Yes.
And I take red localization as the smallest
difference. That is right. Now you have to, this is sort of the simplest way of doing it. And let's call it the null approach. Now what you have to remember is, in my design of my experiment, or in my design of my prediction method, there is one, if you want, flaw. The flaw is that whatever in my, I should have had the, yeah, here's the record. Whatever is annotated here over time changes. Things are added. In the beginning, in the first annotation of this record, I have fewer keywords. Over time, people discover more about a protein, and this becomes more and more complete over time. That means this vector is going to fill up with the ones, or with more ones over time, but initially it will have very few ones, because keywords are missing. And I have to take that into account. So somehow I have to not only actually look at the keyword that I have for my new query protein, I also have to look at the ones where I sort of mutate some of these around. And here we are. So I have to build subvectors where I turn some of these ones into zeros. What if one of the ones in my database in fact had fewer annotations than the one I come with my query? Maybe there should be a one, but there's just no annotation of that, and maybe I find this one in the database, and maybe this is the one that really corresponds to the query. It's just that this annotation here, the one here, is missing, right? So in fact I have to also compute the similarity of subvectors. And that essentially is all that we do here in the system. Then there's no issue about how do I compile similarity, which simply do a Shannon information here. As simple as anything else that you can do. You could have had an edit distance, it's a 1 and 0 vector. We felt that this sort of takes the probability of having zeros into account, and is therefore more meaningful. So essentially it's the information content. And then I simply come in with this query and I match it against the query database. And when I do that, then I discover some correlations that are in fact not trivial. So in this combination here, I know it's cytoplasmic. In this combination, I know, in fact, this is, well, some of those are simple. Actually, they're all simple. Maybe when I wrote those down, I didn't know enough about biology. They're also all simple here, no? This is not really right. This is not really simple at all. This is an interesting combination here. You have a transit peptide and a ribosomal protein, and if you put these two together, you actually have a mitochondrial protein. So this is clearly the type of non-trivial correlation. In fact, any intelligence would put this somewhere else than into the mitochondria. In this particular case, the transit peptide gives it away that at least it can also be a traveler, it can also be in the mitochondria. Anyway, so we have some of these. Actin binding is sort of a giveaway. Coiled cell division is all. Okay, some of them maybe less trivial than others, more trivial than others. And then there are some sort of mistakes. Let's not go into that. Now, we do have a distribution of keywords here. Most of the protein records of the 15,000 we had have between 10 here and 0 keywords. I'm not entirely sure. The 0 is, no, it's not, that's a small number here, it's 150. And I can only imagine that we threw them away. I'm not sure why they appear on this slide here, because we cannot use them. And what I show here is recall precision curve versus number of keywords. So the precision is blue. The more keywords you have, the blue is higher, and then there is a dip here that we cannot explain. Ultimately, this is small data counts that start from there. It's weird that it goes in that direction. It's good that it goes in that direction. Essentially, you assume that this would somehow grow. We cannot establish that, but we can establish it the other way. And that sort of is trivial. The more keywords I require, the fewer things I find. So almost everything has one keyword. This is why here the recall is very high. If I require that I have 2, I have fewer. If I require that I have 3, I have fewer, and then I add. So this is the expected outcome. Now, I will show you some, on my next slide, I will show you some rock type curve where I'm not entirely sure. I'm not seeing the slide. So where it's rock-like, so I'm not going to have numbers here. I'm going to have precision versus recall, and low recall, high precision. So I'm going to have some curve like this. I'm not quite sure how it looks, but something like that. Okay, that means that for some classifications, for very few fraction of my classifications, I achieve a very high accuracy or precision, and if I want to sort of predict everything, my overall level is lower. Okay, now this is the result that I get if I take my top hit. So Natalie said that we find similarity, but we pick the most similar one from the database. So we take the Shannon entropy and we find in all the variations, so removing the ones, so flipping the ones to 0, and all these variations I find the top hit. Now, top hit is the answer here. Say I wondered how good is this thing actually doing for finding the second best. So how would my performance change if I would not only look at the top hit, but the top 2? Does it help to look at the top 2? How would the curve, if the red one is the second best, or the top 2 best, where does the top 2 have to be better than the black, the first hit? So this is, I pick only the top first. If I take the top 2, how does the curve have to look? Where do I place the curve? How does it look? It's not really, I do not even know how this shape looks, but let's assume it looks something like that. But where will the red go? Is everybody clear what I'm doing, or have I lost you successfully? I believe I have lost you successfully. Okay, what I mean by first hit is I go back to this vector the vector assignment here. Try the vectors. Oops. I compare all possible subvectors against my database of vectors. And by the Shannon entropy here, I find the one that is top hit first. With a Shannon score S1. And then I find the second best with a Shannon score of S2. What this curve says is how is the precision versus recall if I only look at the best hit for every single protein. Okay? Now I'm going to plot the curve if I do not look at the top only, but I allow to go in, to sort of take the two best. And I'm not saying the best one is right, I'm saying one of the top two is right. I don't know which one, but I'm allowed to sort of give two to the experimental colleagues that I have. And now I'm going to draw the precision recall curve for top two. Not for the second, for any of the top two. What will this red curve look like in comparison to the black one? So that this is a meaningful thing to do. Do you have? No. Okay. Can red be here? It can. Or what? No, cannot. Why? It's true, it cannot be there. Well, it's certainly not really helpful if it were there.
Because the position is higher than you can see.
So it must be above. Sure. It must be higher. Now my question, if I observe the red to be higher, does it mean that choosing one of the top two is better than choosing only the top? It may or may not be true, but you have to tell me how I find out whether it's true or not. Yes?
You can print a line just for a second. That's it.
And that one would be clearly, if my score works, then, and I'm just going to put that as green. If my score works, then this one has to be below.
Yeah.
Otherwise, my scoring is wrong. If the best score gives my best accuracy or best performance, then the line of the first one has to always be higher than the line of the first. That's true. So it does not answer my question whether it helps or not. Again, I show you that, and I publish in my paper, and you're the reviewer, and I publish in the paper doing one out of the top two is better than picking the top because this is higher. This is why from now on I will never provide to the user the top one, I will provide the top two. That's true. I am chickening out from giving a clear answer. And this is a very, very fair criticism as a reviewer. You can say, this you cannot do because some users will clearly know one answer, so you also have to provide it. That's one stance you can take as a reviewer, and I believe it's a totally fair one, and I believe it's a very, very good point. Yes. There's something else you should come up with. That's a impeccable point. You can clearly make that, and you should make it. So with that, you should induce the one who publishes that into at least providing the score, or at least giving the first, so that users can only look at one. Yes, but there is something else. Okay, let me help you. Do I have a blue? Say this is the top 3. Are you seeing where I'm going? So say I have, and now I misused the green. Anyway, Felix got me off track with this one here. I have 4 states. Well, here's 100% somewhere. This is 4.
It's wrong to only show the precision.
I'm not showing only precision. I'm showing precision and recall. What do you mean? I'm just, so the point is that at some point here precision is 100%. I do not want to indicate that I will draw anything above 100% here. No, but there's something wrong. So this is, say I have a 4-state problem: mitochondria, cytoplasmic, nuclear, and extracellular. So I have 4 classes. And my answer to being right in one of the first top is this. Why don't you jump up? Nobody's jumping up? Yeah?
Yeah, at some point you just include every possible subcellular.
At that point, if I have 4 classes, I do.
Worthless.
That is absolutely worthless. But it's not only worthless. Actually, the green line is below 100%.
Yeah.
Actually, also scary. So it's worthless, and something goes very wrong here. You have to learn as a reviewer to sort of jump at things that are trivial. So this clearly is wrong, right? Okay, for 3 you cannot say that. So let's say we are still at the 3rd, and we're back to the 2nd. Can you see where the argument comes from? Or can you see what you have to... I'm back to the question. Can I say that this one is better than that? Or what do you as a reviewer say? And here's the actual data. So the best, and that is precision, this is the other way around. Precision recall, the other way around of what I drew. That's the top hit, and that's the top 2. Is it better to give 1 out of 2? Red is higher than blue. That's true, but there's something else. What do you have to look at? Yes, Julian?
You have to look at what costs you incur further down if you have several answers that might be correct. So,
what are the costs?
If you read,
what are the costs?
that depends if the researcher...
yeah, that's somehow Natalie's cost. So you're not deciding on one and I'm frustrated. In some sense, yes, you're right, Julian.
No,
that is true. But you don't have anybody to talk to. There is something along the line of what you're thinking. Just do a next step. You're getting in the right direction. Just do it in a more numbered, in a way more counting. Think about how to count, not to talk to your friend in this particular case. Again, I'm always talking about networking. Talk to your friends. It's very, very important. But in this particular case, do some counting. No? Remember the problem when we get to the 4? We're not quite at the 4, we're only at 2. But what is relevant? It's the same kind of idea why the 4 was ridiculous. It's not ridiculous, but there is something we have to look at. Yeah.
Without talking to my friend or the user basically and giving him that to decide what he wants to use, I don't know if it is costly for him to further improve the result with
That's true, but there's something that you have to, as a developer of a method, say about your method. And that has to do is with a question. Let me phrase it in a very impossible to understand way, so that I'm not giving away the answer. It has to do with the question: do you actually add new important information here? Yes?
Yeah, not really. Even if you have a precision of 100%, like 50% of the results are false.
That's the background. So the background is, what are the random odds of what you add? Since if you have 1 in 5 for the first curve, this is 20%, right? If you have 2 in 5, this is 40%. So you have to be at least 20% above the first in order to add important information. Because otherwise you add only through the random signal. So red has to be above blue simply because you change the background probability. One way of doing that is you could simply compute the improvement over random. Okay? So you have to, I would contend, Julian, you have to first make the point that you can show that looking at the first two is better than the random addition over looking at the first. If you cannot establish that, there's no point in talking to an experimental collaborator if what you add is in fact not even better than random. The first hit, I believe is clear, is better than random. But in this particular case, the second one is just barely better than random. But it certainly did not improve enough to really make a big splash of a difference here, in this particular case. But this is essentially what you have to look at. We saw that this is slightly different for different classes. And then we can actually apply it just as the way we did it before to different organisms and see how many proteins we can essentially assign subcellular localization for through these. And ultimately, here we have an image for the entire SWISS-PROT where we remove essentially the annotation and we ask, well, to some extent, for how many of in the SWISS-PROT at the time do we have experimental information? For how many can we do homology-based inference? And then for how many can we do after all of that still this keyword? And you see this is quite a fraction. Well, SWISS-PROT is a biased data set. Now, we can also do that for entire genomes. They are less biased. Yeast, fly, C. elegans, the wheat, and human. And you see again the red one is this keyword based, and you see that in fact the keyword based for all of these organisms is substantial. Substantial only in human, the blue one, homology based inference, is in fact more than red, but for all the others from these keywords you actually learn a lot for inferring subcellular localization. And now we get into the motifs, into this what people typically call the zip codes. And there are two types of motifs that we have. One type is sequence-based and one is sort of conformational. Meaning that the sequence-based motif is I take a stretch-out sequence, I take an amino acid sequence, and I see the sequence motif. There's some motif that stands out, right? While in the conformational motif, the fact is that only upon folding, these two regions come close together. On the three-dimensional structure, I see a binding site, an important motif. But if you roll it out in the sequence, it could be this binds DNA, therefore we have to have a cluster of positively charged residues coming next to each other. But if you roll it out in the sequence, there's a little bit of positive charge here, a little bit of positive charge there. That's nothing special. You find in almost every sequence, right? But coming together, you find something that in fact is a motif. And then you may be aware of the trafficking in the cell. So there are trafficking pathways to begin with. The most important one, or not most important in terms of numbers, but the most clearly distinguished one is the secretory pathway, where we get from the cytosol to the endoplasmic reticulum to the Golgi, sometimes into secretory vesicles, sometimes in the lysosome, endosome, and sometimes on the membrane, and sometimes outside. In fact, most of the proteins that go through this pathway get secreted. This is ultimately one reason why it's called the secretory pathway. The other is that most proteins that get secreted are secreted on this pathway. Then there is a direct gated exchange between nucleus. So Kyla talked about that, between nucleus and cytosol. And then there are sort of some direct exchanges, mitochondria, peroxisomes, ultimately plastids here. So these pathways. Now, if we begin with the secretory pathway, there are so-called signal peptides, and that is work that has been done by Gunnar van Heijnen, who is clearly one of the big players in this field. And the reason, again, I want to repeat why I show these faces and names is to make you aware that you have to network more, but also when you think about literature, you have to think about group authors, main authors. So remember the groups, remember the people who sort of are the major pull in the group, and that one is one of the major pull in the group. Stockholm University, again, this is 8 years ago, H-index 75, is undoubtedly much higher at the moment. So on these slides here, one of the things that you may notice, he announced in 2008, so 10 years ago, the Nobel Prize, because he actually was on the committee of the Nobel Prize for Chemistry. Now, he worked a lot on transmembrane proteins and on signal peptides, going back to the 80s here, publications on the way proteins are translocated, so going through the membrane and then secreted. Some are ending up in the membrane, and some are sort of getting out. And here's another person who is a big name in the field, CERN Brunach. Again, CERN, in fact, I believe has been rising much more than Gunnar over the last 8 years. He now is no longer in Lyngby at the CBS, but he's in the University of Copenhagen, and has moved into medicine mostly. And the work that I talk about here, signal peptide prediction, comes. Gunnar is sort of the expert on the experimental characterization and theoretical characterization of signal peptides. Some of you may know Gunnar from the inside-outside rule, a positive inside rule for membrane proteins. So the way membrane helices are oriented in the membrane is, he discovered how that is done. There's a simple rule: positively charged residues are typically on the outside of the membrane. CERN is the one who brought sort of the machine learning into this field, and Henrik is sort of the go in between these two, who collected a lot of data and expertise from Gunnar, and essentially tried to put it into machine learning device. So the success of signal peptide prediction, or signal P is the method is called, really came from Henrik collecting for many months the motifs or the examples, the experimental annotations, putting that into a database, and then using the prediction method. And looking at all of these data that he collected, he found the following sort of themes. First, signal peptides are N-terminal. Typically 15 to 30 residues. They are cleaved upon translocation across the membrane. They exist in all three kingdoms, and they have a fairly simple architecture. What architecture means, there's an N-terminal region that is described to have often positive charges. A lot of positive charged residues. There's a high aggregation of positively charged residues in the N-terminus. Then there is an H region, which essentially is a hydrophobic region. A region with a lot of hydrophobic residues that typically is over 6 residues, can span to 20 residues, looks very much like a membrane helix. And in fact, most methods that predict membrane helices mispredict this region to be membrane helix. Or in fact, sometimes they are, but let's not get into that. Then there's the C region here that is polar, uncharged, and then there's a cleavage site. Now the cleavage site is the only thing where we can really look at sequence logos, at motifs, for eukaryotes, gram-negative bacteria, gram-positive bacteria, you somehow see immediately in these letter plots there is a lot of information at the cleavage site here. That is -1. Sorry for the quality of the slide. That is 1. So really at this point here, the letters are the highest, the most informative signal for the choice of one particular type of amino acid at that point. And that put into a machine learning device can simply predict the cleavage site. And then this grammar here is put into a device to predict the other regions, and then you can essentially predict whether or not there is a single peptide or where it's cleaved. Okay? Initially that was done with the neural networks, with a neural network, and later it was done by a hidden Markov model. Now, more recently, there again are neural networks and deep learning devices doing that. There are sort of other kinds of things, I will get to the target P in a moment, that have to do with non-classical secretion. That means not using this path, not using the single peptide, getting out differently. And there are many methods out there. This is an example here. This was published, the first method was published here in '97, and essentially this is the method that everybody uses. There's a signal P3 by now, or signal P4, can't remember where they are standing. And essentially all of these other publications are hardly ever used by anybody. Part of that is history, but part of that is really because that group solved the problem best. There is some issue with membrane protein prediction, I already said that, that many membrane protein prediction methods get single peptides wrong, and I'm going to zip over that. Some modern methods, in fact, like Phoebus, Octopus, and others, predict membrane and single peptides at the same time, but I'm not going to go into that. Again, there are many other types of single peptide prediction methods, but there are different types of peptides. So this is what I said, secretory pathway. Transit peptides is the same thing, but for proteins that are transported into the chloroplast. Now, what I have to again point out here is we can train machine learning. These signals, the signal peptides, are such that you can train machine learning devices. There's something common to all of them. There are the rules, the architecture that I showed you on this slide in the, before, but you can really put it into machine learning devices and learn. Okay? And the same is true here for transit peptides. They differ. So signal peptides and transit peptides both are in-terminal, but they are not really picking up the same. So if you run signal P on those, you will not pick them up. But they have similar features. Okay? There's another tool called ChroP for that. There's something called targeting peptides. Again, it's the same thing, but for the import into the mitochondria. The method here is called MitoP. Then there's target P, which sort of joins all three of them, but the sort of more recent version here, I believe, is 4, and that only sort of optimized the signal P. So target P was not updated because of a variety of technical issues. So all of these have in common 3 different types of compartments: mitochondria, chloroplast, secretory pathway, which is not really a compartment, but it's a path. Many of those end up outside extracellular. What they have in common is that they are all N-terminal, and they can all be learned. Okay? Let's move to something that is C-terminal. So there is a retention motif, an endoplasmic reticulum retention motif, Golgi apparatus retention. Retention motif means on the transport in the secretory pathway, so all of these proteins have a single peptide. That means they get into the secretory pathway. Then they get into the endoplasmic reticulum, and some of those are staying there. And some of those that stay there have this C-terminal motif. Something on the other end of the protein that makes them stay there. When you look now at the counts here, what you see is that 91% of the, so these are 2 different motifs here. 92% of the proteins that have this C-terminal motif, in fact, end up in the ER or Golgi. The others don't. So none of these motifs here, for the Golgi retention motif here, the number is 50%. So the point that I want to make is none of these precisions here is anywhere near 100%. So yes, there are these motifs, but these motifs are not such that every one that has this motif is stuck in the Golgi or in the plasmic reticulum. So they are probabilistic motifs, right? So you have a motif, there's some probability that you end up in the endoplasmic reticulum or that you end up in the Golgi, but they are not completely clear. And in fact, they are very difficult to use. At this point, there's not even a data set. So for the retention in Golgi and ER, our group has worked on that for 4 or 5 years, and essentially we have given up. We have built a catalog of all the motifs. We have tried to clean them up, and we didn't find much we tried to predict, and ultimately we have given up. There are examples of publications, but ultimately the problem is they are not really living up. Maybe things have changed since we worked on it, but so C-terminal retention motifs exist, but are not sharp enough to be used for prediction, really. Now let's turn to a different thing that is sharp enough, nuclear localization signals. And nuclear localization signals are clearly sequence-based. There is an NLS, nuclear localization sequence or signal, that is recognized by a protein, such as a protein called importin or transportin. The protein shuttles the protein with the NLS through the gate into the nucleus. One of these, when we started working on this, there was a database of motifs, or there is a database of motifs called ProSite. In ProSite, there is one bipartite motif. What bipartite means is you have a motif here, and then you have a lot of X's. So it can be anything, and then you have a second part of the motif. And the X's, maybe you have 10 or 20 X's, I can't remember how much that was. But so there was some linker region that could be anything, and then there was the motif again. Now, in this particular case, what we found is 96 proteins were known that matched this motif, were known experimentally to be nuclear, and they fell into 31 families. And there was a precision here of 90%, meaning that 10% of the proteins that had that motif, that matched that motif, were experimentally known not to be nuclear. Okay? So, then we went out and tried to see, are there other motifs out there? How would you do that? Well, you would look at a database. There was no database. What would you do then? This is really a simple question. No? Do I need to open the window? Wake up. No? It's not simple? It's totally simple. Just say it. Yeah, how do you do that? So you cannot, when you Google and you look for database, there's nothing existing. So you build the first database. But how do you build it? I'm not talking about which language. Is it MySQL? This is really not relevant. Yeah?
Maybe just look at other proteins that are known to be nuclear, and then check these for motifs that are...
So one idea would be, you create a data set of proteins that are nuclear, and you try to find a motif that they all have in common. And again, we tried exactly that, and in our hands we failed. So there's a lot of software. Meme is one of those examples, but there are many others that for a bunch of proteins, they will simply find whatever they have in common that is a motif. And failed for us meant we would call success only if we could sort of twist this into being 100% right. So we wanted something that is only found in nuclear proteins. So it would not match in other proteins. So what we did is we sort of had a set of nuclear proteins, and then we found nothing that is common. So we sort of shrank it. We looked at subset of these. That's another thing. We want the motif to span between families. We want the motif to be something that is also found between proteins that I cannot just do homology-based inference for. I want to find something that goes beyond families, right? And that ultimately, somewhere in between, we lost the signal. But that's one approach. There's something else. I mean, how do you build your database? So what Daniel said is a completely in silico approach. And in fact, we tried that. because we are lazy. If that would work, that would be the best way of doing it. Because you can apply to anything, right? You can build arbitrarily large. If that method really worked, you could build very large data sets. That's the beauty of that. And very quickly, all you need to do is program a little bit. But if that doesn't work, what do? The answer is really simple. Just say it. Well, you look at the literature. You read and read and read. So you look at everybody who is sort of describing any. You look for the keyword NAS, nuclear localization signal, experimental characterization of motifs, and then read hundreds of papers. And this is actually what Murat Çokol really did. And then he sort of aggregated some of these motifs. Here are the quotes of the papers, and you already see that in some papers there is more than one motif mentioned. Sometimes they are similar, sometimes they are not. So Murat also was, that means that Murat was also clever enough to find papers that tended to discuss not single motifs, but sort of a bunch of motifs, so reviews, that is. And red means positively charged residues, so you immediately see that there is a lot of red on these motifs. You also immediately see that they're all different length. Some of them are actually extremely short, and just to show you some more, so this is one that is a bipartite motif with many X's in between here, and some of them are really, really, really short. And now all of these motifs are such that the experimental group said, if I take this part out, I can no longer see that the protein is in the nucleus. Okay? Does it mean that it's the right motif? So they work on a protein. In that particular protein, they establish that protein is nuclear. They sort of modify these residues here. And when they modify those residues, they see the protein no longer is nuclear. Hence, that is the nuclear localization signal. They modify other residues too, and then they still see it in the nucleus, yes?
Wouldn't it have a different fold after being modified?
Yes, so there could be, typically what they do an alanine scan. So meaning they take only one residue out and they mutate it to an alanine. And they hope that that will not have a different fold. So at the moment you sort of completely change the fold, you mess up. And ultimately what you will see, so if the answer to your question would be yes, so if you change so much that you would actually change the structure, then you will no longer see it in the nucleus, right? So then you will suddenly see it as something that no longer is there. But then you would argue, if that happened by sort of modifying that motif, then that motif is relevant for it. In that particular case, you would say it's not really the motif, you change the structure, right? Fair enough, that is one criticism, yes. But what else could be an issue here? Yes?
They only tested for alanine replacement.
They typically only have alanine replacement, yes.
And then it could be that another amino acid that is similar will still work.
That is true. That is true what you say. That is true. But I take that point. But my question is on, you're completely right. My question is sort of on the other side of the spectrum. The point that I'm trying to make is the fact that changing that particular sequence here in that particular protein does not mean it's a nuclear localization signal, because you may still have the same thing in a protein that is not nuclear. Right? So that's what you have to actually look up. You have to actually, for all the motifs, you have to build a data set of proteins that are nuclear, experimentally known to be nuclear, and a data set of proteins that are known to be not nuclear. And for whatever is in the experimental article claimed to be a nuclear localization signal, it cannot be a nuclear localization signal if it matches also here. Right? So you're right, Julian, but this is sort of the more crucial problem that you have, right? Now, say I gave you the opportunity to sort of change the motif. So maybe this motif sort of is around something that is relevant. but maybe it's too short. What would I do? I would make it longer. How would I make it longer? And then, so I would align the protein, I would align I have my query. What was it, Harned? And then I have my homolog 1 here, that is H, R, A, R, and T. And then I have homolog 2, it's H, A, R, D, and T. And then?
The residues before and after.
Yes. Now what would I do? It's totally simple, just say it. Or did I, huh?
You already added
A P, yeah.
at the very.
So, but this is not my, okay. So this is my sequence alignment, right? Yes, and you say I already added it to the motif. Misunderstanding. So here's my alignment. But yes, so the idea is exactly that you look at extending left and right according to ideally what is conserved in the alignment, and then you choose which side of the Harned is actually conserved. So if left you have a P that is conserved, you extend into the left side, and you extend as long as you find some conservation. And you do that, so you do it one by one. And you always ask, am I becoming specific enough? So the moment the P, H, A, R, and T is such that it does not map in any protein that is non-nuclear, I say enough. Okay? There is another issue. I could make it longer and longer and longer. It would become more and more specific. So when do I have to stop? And I stop, ideally, at a point where I demand that at least two different families of nuclear proteins have to have that motif. Because otherwise I'm too specific. Otherwise I'm actually ending up writing the sequence of Q there, not the localization signal, right? Because I want something that is shared between families. There is this thing that I didn't tell you yet. So these nuclear localization signals are really so extreme that you can put an NLS upon gold. And gold, I really mean gold, not the protein gold, but a piece of gold. And the piece of gold is imported into the nucleus. That extreme they are. You just have this tag, this little tag, you engineer it. So somebody did that experiment. And so we sort of iterated through this. When they were too short, we made them longer. And when they were too long, here's an example for that, then we made them shorter, exactly with the same thing. Looking at the sequence alignment and picking and seeing that in this iteration here, we improved the motif. And then we came up with a set of 214 motifs that in those days mapped in 1,300 proteins. By design, simply because we always assumed that none of the proteins that we know is not nuclear has the motif, it must be 100% here. This is a design issue, and we sort of mapped 40% of all the then known nuclear proteins. It turned out that over time, so this was done in 2000, we just republished that last year, and it sort of had fallen in terms of precision and in terms of recall, and we sort of had to refine it. But essentially this is the way to do it with the nuclear localization signal. The important story here is the NLS, again, that comes from looking at these, you see that you cannot, or you begin to, bless you, that you begin to suspect that you can actually not machine learn this. This is not the type of architecture that you found in the signal peptide. The only thing you really, at least so far, the only thing we found that we can do so far is archive those motifs. And make this archive as big as possible, possibly somehow add Daniel's ideas or add some sort of in silico motifs. And in some sense, this iteration here was a sort of in silico expert-driven kind of technology, where we got from the 91 we found in the literature to sort of expanding them into 214. that is sort of a thing in between the two. But it's clearly something that you cannot do by machine learning, okay? This is a different type of motif. Then we sort of ran that on genomes, and we found that we found many, but in fact we still found only a limited number of all the known proteins. We discovered new motifs, and then we found something that we found very interesting. So we did plot. We just visual for the publication. We want to visualize some of the motifs, and then we saw this. What you see in red and orange, so in the space filling model here, is the NLS. Comment. What does it mean, what you see? Can anybody comment?
Is the orange part the motif?
No. What do you believe the orange one is?
The DNA.
Yeah. So?
It's the DNA binding site.
Yeah. So we have an NLS, that is a DNA binding site. That sounds as if you bind to the nuclear localization signal, the importin binds to the nuclear localization signal, it gets into the nucleus, then the importin goes off, the NLS is free, it binds DNA. And that in fact is what many proteins in the nucleus do. So you simply reuse the same binding site. And that's a totally cool mechanism. And it turns out there is in fact even another iteration. There are some export signals. So-called NES, nuclear export signals. And some of those, some of the types of proteins that export, some of them are called exportins, for instance, they bind the same signal again. So you bind the importin, get into the nucleus, importin goes off, you bind DNA, DNA goes off, exportin comes and takes it out, and you have a cycle. A very simple cycle, always using the same motif. It's not true for all nuclearization signals, but for some of them it's exactly that. And it's a beautiful, simple model. So it's, in some sense, it's the one moment in computational biology that I went through where something was really simple. And that allowed you to somehow actually also from these nuclear localization signals predict DNA binding. Overall, in terms of the numbers that we find, the analysis is not shown. We are just back to what I showed you before. Okay, now clearly what we see here is at the end of the day there is still a lot of white. Nuclear localization signals, signal peptides, experimentally known localization, homology-based inference still does not cover everything. So we do need to predict de novo, and that brings us back to the first question. And that's sort of the zip code story from Günter Blobel, who got the Nobel Prize for this entire idea, that you have a zip code that leads to the way proteins are transported through the cell. So, what we know today, and I call them signals, motifs, zip code, whatever you put in there, for most proteins, they're not known today. Why? One thing could be they remain to be discovered, right? Or maybe there are other types of motifs. The other type of motifs could be the type of conformational motifs that are difficult to find, because for most proteins we also don't have the structure. So if they would all be this type here, it would be really difficult to discover them. In fact, the fact is that typically for proteins for which we know structures, we know so much more that people don't really look at the localization motif at them. People have not really gone down the path a lot, so there are not many of these conformational motifs that are known. Another reason why maybe many of those we don't know is there are other mechanisms. And that's my question to you. What could be this other mechanism? And that's what we discussed just now, right? So this other mechanism is the cargo mechanism. So you have a protein with a zip code, so nuclear localization signal, recognized by importin. That protein happens to bind to the green one. The green one is exactly what I said before. It does not have a sequence feature, neither length, no, so something that really is nondescript. for at least the process of importing into the nucleus. Yes, maybe there's a length preference, back to what you said, Kyla, but maybe not, because maybe just this green one is binding to a protein that is imported into the nucleus, right? And it sort of gets pulled through with it. And what we in fact somehow would have to do, no, I want to go further today. At some point, maybe when we get closer to Christmas, maybe we should just all sit down and think about, can we, I never had enough time to think this through. Maybe there's a way in which we can sort of estimate how much that could contribute. So saying for the nuclear localization signal, for instance, at the moment, I don't know what the coverage is. Maybe we can, for 30% of all the nuclear proteins, we know the localization signal, right? So for 70%, we don't. Can we sort of gauge, estimate how many of these green cases we have? And I'd like to think through. For the time being, what we clearly need to do is de novo prediction. Again, de novo prediction means, in contrast to for homology-based inference, now I'm going to machine learn something that is common to a bunch of proteins, and that allows me to predict subcellular localization. In the simplest way, we predict in 3 different classes: cytoplasmic, nuclear, and extracellular. And in the simplest way, we already talked about that on the whiteboard, there's some correlation between amino acid composition and subcellular localization. That was initially established by Kenta Nakai and Minoru Kanehisa. Do you guys know Minoru Kanehisa for anything? The name Kanehisa? So I'd argue that you all have used a database that Minoru created. Guess what that database is. I hope it's not written. No, it cannot be written. Anyway, so the initial work simply established the correlation between amino acid composition and subcellular localization. Kanehisa. Nobody? So the database starts with a K. Yeah. Which is a tremendous achievement. Cake, essentially, around about the same time, Minoru started to begin putting all proteins into cake and manually annotating cake, one by one. And people really believed that it was a daunting task that could never be done. 30 years later, we have this resource that is invaluable. Anyway, my question to you, there's a correlation between amino acid composition and subcellular localization. Do you believe this? Okay, again, it's a publication, and publications you have to believe. No, not really. You can still doubt them. In this particular case, this is not what I'm after. I'm not after saying Kanehisa is wrong. What I'm trying to say here is in your biophysical understanding of the problem, why would amino acid composition correlate with subcellular localization? Yeah.
In different compartments, and maybe some proteins with better.
Yes, so you have different pH values, you have different environments, and you may very well imagine, since amino acids have different biophysical features, that in some environments you have more charge, and some you have more of this, and some more of that, right? So that the overall amino acid composition would differ. Yes, that would exactly be the explanation, and this is in fact. And I apologize for the quality of these slides, which in fact is why I forgot to switch on the lights. So what I show here is a principal component analysis of 3 types of proteins. Light green, that you may sort of make out here, is nuclear. Blue, extracellular. Red is cytoplasmic. And the principal component, the first two eigenvalues here. So essentially I'm looking at the eigenvalues that best separate these proteins into these three classes. And you see some separation. The separation is upon amino acid composition. Okay? You see, again, I'm sorry for the quality you don't quite see, but at least you would not jump up if I told you that there's some blue cluster, some red cluster, and some green cluster. Okay. Now here, no. Has anybody read it? The next one? No. So there's a different pH value. What part of the protein sees the pH?
The outside of the protein.
So what would you plot to make that point?
The charge of the outside amino acids of the protein.
Yes, you could do the charge. If you believe the charge is what it is. So I'll show you this. I cannot read them. This could be a charge. This looks like an R. So charge clearly has, even if I cannot read it, I know charge clearly has an effect here. So one of the principal components most likely has at least one charge in there. The second one I cannot read at all. Also not on my computer, I guess. Anyway, but it's not only charge. So what do you have to do? What? Is that difficult or is it trivial? Difficult. You have to do the same thing. A principal component analysis, not just charge. You do, in fact, the best thing to do is you do surface residues and core residues. So these two together are that. Okay, let's begin with the core residues. In the core residues, so now you see green here, you see green here, you see green here, you see green here, 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 and here. Blue, 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 here. Red, essentially mostly here. So core, yes, there is an N that is sort of different from all of those, but there is N that is in the middle. Clearly the inside residues are not able to distinguish between blue and red. At least that is what you can say. And clearly, what you can also say is this one is a much worse distinction than this. Conversely, what you can see here, when you look only at the surface residues, again, now is the green ones here. Here is sort of the red cluster. There is green, there's green here. But still the majority is red here, and then the blue here is completely separate. So there's no blue in the red, such as here, or even as here. So clearly, if you visually picked the one that distinguishes best, you would immediately put this. If you picked the one that distinguishes the worst, you would pick this. And then you, in fact, have verified this suspicion that in the amino acid composition, it's not really amino acid composition that counts, it's only the surface, the outside of the protein, which makes a lot of sense because only the outside sees different pH values, right? And by the way, when we looked at some of those that are confused here you see there's a blue in the red. We saw that these were glycosylated. So you put essentially a sugar on your protein to sort of change its biophysical features. You have a protein that sort of sits in the wrong space some of its lifetime to simply, and you do that by glycosylation. This in fact is one effect of glycosylation. So these are the glycosylated ones here. And they are in fact the most sort of confused ones. When you average over these three classes, do a simple percentage of amino acids, you immediately see in a letter plot that the three classes clearly differ in terms of these letter plots. And that brings me to the problem of predicting solvent accessibility. Because in order to do this in a prediction system, first you have to predict solvent accessibility. And solvent accessibility you essentially can predict. So the way you define solvent accessibility, you roll a ball of water over the three-dimensional structure, and then you compute what fraction of the amino acid is available to water. And then there are different states of amino acid of solvent accessibility. One sort of completely not accessible. And not completely not accessible, you would count C and D. A is a little bit accessible, B, E, F, and G, they're all a little bit accessible to water. And you could count them surface accessible, and C and D are non-accessible, so it's a binary state. You can train a machine learning neural network in this particular case that uses evolutionary information to predict. Here it's not in a binary state, but you essentially predict the percentage, and in this particular case you predict the percentage as a square root of a relative percentage. Let's not get into that. Overall, there's about 75% accuracy in a two-state prediction. And you can use those predictions to, for instance, label proteins that is conserve and consec from Nirbental. Here, that's a method. Here, conserve, it just takes a three-dimensional structure, takes an alignment, and labels on the three-dimensional structure everything in dark red here that is conserved, versus everything that is not conserved at all in sort of really blue. So here's a blue, here's a blue. And what you immediately see jumping at you is the binding site. So the more conserved you are, the more you really, on the surface, the more you're really in the binding site. And consec, in fact, is a method that does the same thing from sequence. And that is a tool, whenever you look, whenever you have 3D information available or only sequence information available, you should really look at conserve and consec. By the way, there's also a great book from Nirbental on proteins. Has anybody seen that book? Second edition? Proteins, protein structure. I should look at it.
Professor Frischmann.
Say it again?
Professor Frischmann has shown this.
Has shown you the book.
Yeah.
That puts you off. Which one? The big one?
No, he only shows the cover and some examples of some of the protein structures.
Okay. So if I show you the book, then you do not necessarily feel like looking at it. So the first edition was already 600 pages. The second edition is substantially bigger. But it's a great book. Just look at it. Okay, then we go into prediction, de novo prediction of solvent accessibility. So essentially one thing to say is this is a de novo prediction, the PCA. It's just we would have to define the first principle and components, and then would define some threshold, ideally here, and then we can have a classification, right? That is some de novo prediction. It's not machine learning. It's much simpler. It's a PCA, and the work in this particular case has been done by Rajesh Nair. And Rajesh, so this was published here in '98. Rajesh redid this in 2002, and now. This is surface, this is amino acid composition for the whole thing. The colors have changed. Green is still nuclear, red now is extracellular, and black is cytoplasmic. So what you immediately see is that none of these classifications is as good as it was shown before. So essentially 4 years later, the database had grown so much that the signal that we initially published was no longer as clean. And in fact, we still felt that this is better than this, so you can compute it, it's not felt, you can just put numbers to this. But it's also clear from this image here that PCA is not enough. We have some information in there, but we do need to learn this by machine learning. And that brings us to the concept of neural networks. So neural networks essentially are devices that have an input here, an output. There's a connection between the input and the output. The way the device works is the input has a value. You multiply this value times the connection here. At this point, you add the multiplication this times that, plus this times that. That's what comes in here. In a geometrical interpretation, this is multiplication of two vectors, the vector of the input units and the vector spanned by the connections here upon one another. And then you, for what comes out here, you apply a trigger function, such as the hyperbolical tangents here.